So this is one of the competencies that is in your syllabus, that is the structure, types, derivatives, functions, okay, normal range, everything in your syllabus. Okay, so we'll try to cover this hemoglobin section in theory today, then we'll do the practical work in the next week in the laboratory, okay, by your own blood. So be prepared, get the lancet, okay, so that we can proceed for the practical purpose. Now, where the hemoglobin is present and how is it getting formed in the RBCs. RBCs are the erythrocytes, okay. In a mature RBC, the cell organelles, they are missing. Mainly the cytoplasm comprises of the hemoglobin. Now, at what stage of the RBC the hemoglobin starts getting formed is the intermediate normoblast, that we will see in the next class when we study the erythropoiesis or the formation of RBC. Okay, poiesis means formation. Okay, step by step we will see the poiesis of RBC, WBC, platelets. Okay, when we are talking of formation of RBC, okay, it is erythropoiesis. It has number of stages, and one of the stage in which the hemoglobin formation starts appearing is the intermediate normoblast. Okay, so what is hemoglobin? Is a protein. Again, it is a conjugated protein which is present in the red blood cells that binds to the oxygen, give the oxygen to the tissues, okay, and carry carbon dioxide back to the lungs. This is a simple way to explain what do you mean by hemoglobin. Okay, where it is present, the first and foremost question which is asked in your practical viva is, where is hemoglobin present? The first and foremost answer, 90%. With time, the common sense or the IQ level slightly decreases of the first MBBS student. Okay, again it has to take a steep rise. So it is a graph which is followed normally, okay, every year. So they are present, the first answer comes in blood, so it is present in the RBC. It is correct, but you have to specify the answer. It is present in the red blood cells. Okay. Now, okay, structure and function. What is the structure of hemoglobin and what are the functions of hemoglobin? It is a conjugated protein, okay. Mainly when we divide the term, it is divided into heme and globin section. Okay, just try to understand, then you just write it down. Okay, this heme section and globin section. So the question may be asked, write the biosynthesis of hemoglobin, okay, or the formation of hemoglobin. So the word hemoglobin, when it starts appearing from the intermediate normoblast, that is the stage, one of the stage of erythropoiesis in which the hemoglobin appears for the first time. Now this heme and globin, okay, heme is made up of what? It is a protoporphyrin 9 structure, okay, and this protoporphyrin 9 structure when combines with iron, and which form of iron? It is the ferrous form of iron. When it combines or joins with the ferrous form of iron, it results into a structure that is known as heme. Okay, so a vague idea is heme and globin. Exclusively when we are talking of heme, it is made up of the combination of pyrrole rings. Okay, now these pyrrole rings join with the iron. Which type of iron? It is the ferrous +2 state of the iron that is only usable for the formation of hemoglobin, that will function in most 100% manner. Okay, so when this protoporphyrin 9 structure combines with iron which is present in the ferrous form +2 form, results into the formation of heme. We have broken the word hemoglobin into two parts, heme and globin. When we are talking of heme exclusively, what are the parts or what are the structures which are involved for the formation of heme, and when we are talking of globin, what are the requirements or dietary supplement required for the protein chain formation. So this class is horizontally integrated with biochemistry also. Clear? So the same thing can be asked in biochemistry also, but the applied aspect can be asked in the physiological section also. Okay. Normal hemoglobin consists of 4 heme groups which contain a protoporphyrin ring plus iron. So heme is an iron containing protoporphyrin 9 and the combination of globin. Okay. And what is globin? It is a tetramer of 2 pair of polypeptide chains. Come first, the iron must be in the ferrous form and the molecular weight is 68,000. In some books it might be 68.4. Specifically when we are talking, it is a tetramer. Okay, so each and exclusively single chain, globin plus heme, okay, it comprises of 16,000 kilodalton. So when combination of 4 chains, it results into 68,000. That is the structural weight of hemoglobin. Okay, a protein called globin made up of 2 alpha and 2 beta chains. Okay, in case of heme, it was the pyrrole ring, the combination of 4 pyrrole resulting into tetrapyrrole, resulting with the protoporphyrin ring plus iron present in the ferrous form, resulting into the heme structure. Okay, now we have to think or study something regarding the globin or the protein chains. When we are talking of the protein chains, they are broadly categorized into alpha and beta. Okay, other than beta, they have the delta, gamma, epsilon, different varieties of chains. Alpha is the most common chain, 2 alpha chains. Okay, their sequence of arrangement of amino acids remains same in the alpha, and it is 141 amino acids which are present in the alpha chain. That is, remain the same. The only variation comes in the beta, gamma, delta chain, but their number of amino acids generally remains same, that is 146. On the basis of the changes seen in the different globin or the protein chains, the different terms or different types of hemoglobin are seen. That is the adult hemoglobin A1, glycated hemoglobin, fetal hemoglobin. These are the different types of hemoglobin. When you just read the paper or just listen to the questions in the Viva, okay, be very much specific what is asked. If they are asking for different types of hemoglobin or whether they are asking for different derivatives of hemoglobin. Okay, structure of hemoglobin, then comes types of hemoglobin, then comes derivatives of hemoglobin. So there is difference between the types of hemoglobin and derivatives of hemoglobin. Clear? Types and derivatives. When we are talking of types of hemoglobin, what do you mean by types of hemoglobin? There will be change in the nature of the second chain. First is the alpha, second may be gamma, beta, delta. Okay, on that basis, the second chain basis, the hemoglobins, they are classified into different types. Clear? And derivatives, which we will see later on, though they are the different things. ठीक है, तो वाइवा में जब भी कोई क्वेश्चन आपसे पूछा जाए या आपके सब्जेक्टिव नॉलेज के असेस किया जाए, प्लीज बी वेरी मच स्पेसिफिक टू द क्वेश्चन विच इज बीइंग आस्क्ड, है ना, कि हमें ये नहीं लिखना है कि जो हमें आता है, जो पूछा गया है वो आपको बताना है. क्लियर? Protein called globin made up of 2 heme molecule group bears an atom of iron which can bind to 1 oxygen molecule. Each hemoglobin molecule thus can transport 4 molecules of oxygen. Okay, so each molecule of each hemoglobin can carry how much oxygen? 4 molecules of oxygen. This is the structure. We can see the 4 chains present: alpha 1, beta 1. Okay, the top one is the beta 1, beta 2, alpha 2, alpha 1. This is the heme structure which is attached to the chain globin chains. Okay, this is the detailed structure. You can see the 4 pyrrole structures. N molecule of this is binded with the iron which is present in the ferrous form. Okay, now this iron, this can bind with the oxygen. It has a special affinity to bind with oxygen. Okay, this is the structure of the heme, and this is the your globin structure. Totally comprising is the hemoglobin structure, iron containing heme group. This may be asked in biochemistry. It can also be asked in physiology. It has an integration. Okay, the iron has to be present in +2 form. If it is not present in the Fe2 plus form, it will not be able to bound with oxygen. Okay, most important part. When we are talking of oxygen, the iron part or the +2 state is responsible for binding with oxygen. When we are talking of carbon dioxide, another function of hemoglobin is, first function is it transports oxygen. Second function is it transports carbon dioxide. When we are talking of transport of carbon dioxide, it is mainly responsible, or it is mainly done by the globin chain. Okay, when we are talking of ferrous, you have to keep in mind the ferrous is highly responsible for the combination with oxygen molecule. We have to keep in mind that there are 4 hemes attached to 4 different globin chains. Each heme has a molecule of, each heme has a iron +2 which can combine with oxygen molecule. Okay, इसको दिमाग में रखते हुए अपन इसके आगे की प्रॉपर्टीज पढ़ेंगे. इसलिए इस स्ट्रक्चर को अभी एटलीस्ट एक विजुअल इम्पैक्ट हमें याद रखना है ताकि हम इसमें रेस्पिरेटरी सिस्टम तक इसको रिकॉल कर सके. इस बेसिस पे इसका सबसे इम्पोर्टेंट फंक्शन आ जाता है, दैट इज ऑक्सीजन डिसोसिएशन कर्व. So this oxygen dissociation curve you will study in respiratory system, one of the important topic on which either you get a lengthy question or a short note definitely. Clear? So it is not that this will be over in this blood or blood topic, it will be covered later on in the respiratory system. तो वहाँ जब respiratory system होगा तब ये नहीं कहना कि हमें तो पढ़ाया ही नहीं या हमें तो बताया ही नहीं. इसको आपको कोरिलेट करना पड़ेगा कि ये कैसे इसके फंक्शन को इन्फ्लुएंस कर रहा है. So this is the structure. Heme is protoporphyrin. In some books it is written protoporphyrin 9 plus iron. So need not to get confused. You may write protoporphyrin plus iron or protoporphyrin 9 plus iron. Okay. So it is a tetrapyrrole and pyrrole ring joined together by 4 methine bridges. That is mainly covered in your biochemical sections. We are not more concerned with this. We are concerned with the function, but you can easily see the attachment of the iron with the nitrogen formed. Now how it is getting formed? Now normal hemoglobin production, what are the 3 requirements for the hemoglobin production is? Most abundant and most important requirement is iron. Which type of anemia is most common in India? It is iron deficient anemia which is most common in Indian population or Indian scenario. Okay. Why there is requirement of iron? Okay, in pregnancy. Pregnancy is a normal physiological process, aging process, normal physiological process. Okay, normal hemoglobin production is dependent upon 3. First is the adequate iron delivery and supply. The supply has to be regular. Okay, though the supply may be normal, but the body may not be able to absorb or utilize the iron. Okay. The iron which is present in the dietary form or which is taken in the dietary supplement has to be absorbed. The site of iron absorption is small intestine, duodenum and jejunum. That is the site where the iron absorption takes place. For the iron absorption to take place, we require vitamin C. What is the role of vitamin C? Caspase intrinsic factor. Hmm? No, no. Vitamin C, cobalt, copper, and certain minerals B6, they are required for the absorption of iron and the further stages of erythropoiesis. In case of erythropoiesis, when we will take the next class, we will see their different roles. Today we are concerned mainly with the hemoglobin, but they are required for the synthesis of hemoglobin. Okay. Now the next comes the adequate synthesis of protoporphyrins. Okay. Porphyrin rings, spirals, from where they are getting formed is the combination of glycine and succinyl coenzyme. Okay. Iron content of hemoglobin and adequate globin synthesis. For globin synthesis, what is required is proteins. Okay. What are required is proteins. So main requirement is the iron, protein, porphyrin ring structure formation. Clear? These are the requirements for the formation of hemoglobin. So this is the content of hemoglobin, 0.33%. So if this is the requirement, 100 ml of blood containing 15 gram of hemoglobin contains approximately this much of iron. Okay. Now in our practical class next week, we'll do hemoglobin estimation of your own blood. Okay. So we'll take a sample of 20 microliter and we'll do the calculation and you'll do the experiment and come out with the value of your own hemoglobin concentration. Clear? This is an index. Okay. Hemoglobin has an important function of oxygen binding. So 1 gram hemoglobin binds with 1.34 ml of oxygen. So with this information, we can calculate the oxygen carrying capacity of your own blood. If a student or a subject has a hemoglobin of 10 gram %, then how much oxygen carrying capacity he has? 10 multiplied by 1.34 ml of oxygen. So, हंसी आ रही है या बोर हो रहे हो? आ रही है? क्या कि लंच सेशन के बाद में बहुत मुश्किल होता है अवेक रखना? So this much of oxygen carrying capacity of a student, a subject. Okay, that is the result of your experiment. Okay, but ideally when we are talking, it is 21 ml is of male, adult male, and 18.5 is of adult female, the standard oxygen carrying capacity. Yeah. So we have to correlate theory and practical simultaneously. This is the hemoglobin synthesis. Now you have to remember important thing, in case of hemoglobin synthesis, the site of heme synthesis and globin synthesis, where they are taking place in the erythroid series cells, that is the normoblast, starting from the intermediate normoblast. When we are talking of blast, okay, we'll see in the erythroid series, but the synthesis of heme takes place in the mitochondria, whereas the synthesis of globin takes place in the under the ribosomes. We have initially broken down hemoglobin into heme and globin. When we are talking of heme, where the synthesis is taking place? In the mitochondria. When we are talking of globin, they are taking place with the help of ribosomes, that is in the cytoplasm. These are the two main sites where the hemoglobin starts its formation in the intermediate normoblast. ये सबसे common MCQ आता है कि where does the hemoglobin start appearing for the first time in the erythroid series? Only in your exams, but later in your competitive exams too. So according to Guyton, it is intermediate normoblast. Whenever you are answering any MCQ, anything, please follow Guyton or Gallo. Clear? So you can see the structure here, the heme synthesis taking place in the mitochondria. Heme is coming into the cytoplasm, and there the combination with the globin resulting into the formation of hemoglobin. You can see another structure, that is Fe plus 3 plus ferritin. If you remember, in the plasma protein we have talked about apoferritin, transferritin. These were the plasma proteins of which nature? Globulins, if you remember the class. Okay. So on the basis of these globulins, is transported and it is kept and is recycled in the body. Okay, one of the form is ferritin, another form is hemosiderin. So there is difference in the concentration of iron in the ferritin and hemosiderin, another form of plasma protein. But this is the whole structure where we come into information or clarity that heme is getting formed in the mitochondria and globin part is getting manufactured and formed in the cytoplasm of the cell. Of which cell we are talking of? It is the cells of the erythroid series starting from the intermediate normoblast. Clear? Now, what are the factors which will affect the synthesis or the formation of hemoglobin? Is the role of proteins, role of iron, role of other metals like copper, cobalt, calcium, etc., and role of vitamin C, vitamin B6, folic acid, responsible for the formation of nucleus and for the erythropoiesis to take place or for the further formation of RBC. These are the basic requirements. And in case of erythropoiesis, when we are talking of the formation of RBC. And a mature RBC has what? Maximum part of the RBC is covered with hemoglobin. Okay, so these are required in safe quantity, but they have their roles. These are the derivatives of hemoglobin. Initially, we have talked about types of hemoglobin: adult hemoglobin, A1 hemoglobin, glycated hemoglobin. Okay, these were the different types of hemoglobin, fetal hemoglobin. Okay, in which there was difference in the second chain. One is alpha, the other beta, gamma, delta, epsilon. The change may vary, but these are the derivatives. When the hemoglobin combines with oxygen, it is oxyhemoglobin. Okay, so when the question comes, write down the or enlist the different derivatives of hemoglobin, you have to give this. When it is asked enlist the different types of hemoglobin, then the answer is different. Okay, oxyhemoglobin combination with oxygen. Now when we are talking of combining with the oxygen, as we know, it has a 2 alpha, 2 beta chain. Each chain has a heme has iron present as ferrous. Now ferrous has a special binding affinity for oxygen. It has neither too less affinity, neither too high affinity for oxygen, because if it has too less, then also there will be resulting into pathological condition. If it has too more, so it will not be suitable for exchange, easy exchange of gases. Now once the oxygen combines in either alpha 1 chain, suppose, okay, it will give a beneficial effect and the other alpha 2 chain will automatically get faster combination with oxygen. So it is a heme type of interaction. Okay, एक ऑक्सीजन मॉलिक्यूल मेरा तो उससे स्पीड अप होके सेकंड वाला उससे भी फास्ट ऑक्सीजन के साथ कंबाइन होगा, तीसरा फास्ट कॉम्बिनेशन कंबाइन करेगा, चौथा उससे भी फास्ट करेगा. दैट इज नोन एज heme interaction. In some books you might get, in some you might not get it, but this is just an extra information which I am giving you. That is the role of how the oxygen gets bind easily with hemoglobin. So it is responsible, or one of the chain has a initiating step and that is followed by the other globin chain. Okay, so it is heme interaction, and the heme C+2 has a special affinity for binding with oxygen. That special affinity depends number of factors. ठीक है, उसकी एक affinity तो है कि वो ऑक्सीजन के साथ बाइंड करेगी within a fraction of seconds, less than a second. Okay, within a less than of a fraction of second the oxygen binds, but this binding of iron with oxygen. इतने कम टाइम में क्या होता है, लंग्स के थ्रू जब कैपिलरीज वहां पे पहुंच रहा है ब्लड, देन इट विल इमीडिएटली कैप्चर द ऑक्सीजन. It has to capture the oxygen because the blood which is reaching to the lungs, okay, the capillaries which are reaching to the lungs, they are not oxyhemoglobin, they are not oxygenated. They will catch the oxygen, they will carry the oxygen. That oxygen has to be given to different parts of the body. Okay, so when this is carried, this is rich in oxygen, it will be run and carried and given to the other areas of body immediately. Okay, so it has a limiting step. Now this association and dissociation of oxygen, okay, so this in respiratory system when we see will show us a sigmoid shape of graph. When the partial pressure of oxygen is 100%, then how much oxygen is carried by the hemoglobin, that comes out to be 100% saturation. Okay, so when the calculation is done, round about 15 gram percentage is considered to be 100% saturated with oxygen. Okay, so in laboratory when you will come for the experiment, the tube has two readings or two scales. अपनी छोटी स्केल होती है ना 1 सेंटीमीटर 1 इंच की, वैसे ही वहाँ पे जो ट्यूब होगी उसमें दो रीडिंग्स होती है, 1 ग्राम परसेंटेज की और 1 परसेंटेज की. So if she does the experiment and her hemoglobin comes out to be suppose 9 gram percent, so 9 gram percent is equivalent to how much percentage? So simultaneously you have two scales, one is gram percent, another is percentage. So the examiner or the faculty or the teacher may ask is the percentage, what is the gram percentage? So you have two scales, two units present on the tube. Okay, so your observation has to be very sure that in what scale you have to give your answer. Scale is asking, ठीक है, कि वो ग्राम परसेंटेज में पूछ रहे हैं या परसेंटेज में पूछ रहे हैं, या वो दोनों में पूछ सकते हैं कि अगर आपका इतना ग्राम परसेंटेज है तो वो कितनी परसेंटेज है. ठीक है, तो ये ऑक्सीजन डिसोसिएशन कर्व अपन पढ़ते हैं कि ऑक्सीजन डिसोसिएट किस लेवल पे जाता है और कौन कौन से फैक्टर्स होते हैं जो कि इसकी शिफ्ट के लिए रिस्पॉन्सिबल होते हैं, शिफ्ट टुवर्ड्स राइट और शिफ्ट टुवर्ड्स लेफ्ट. Shift towards right means easily it will dissociate and easily it will give. So whenever you require or you perform any exercise or any activity in which oxygen requirement is moved, खड़े हो जाओ थोड़ी देर, अब तुमसे पीछे खड़े हो जाओ थोड़ी देर, अपने आप नींद चली. Hope the lecture is not that much boring. नहीं हो तब भी झेलना तो पड़ेगा ही अपने को, ठीक है ना. सॉरी करने से क्या नींद नहीं आएगी? सॉरी मतलब मैं दोबारा नहीं करूंगा, तो सॉरी करने नहीं आएगी. क्या नहीं, खड़े रहो 2 मिनट, तब तो वास्तव में नहीं आएगी, है ना. Oxygen hemoglobin is oxygen combining with hemoglobin, so it has the functions which will do a detail in respiratory system. तो उसको उस टाइम पे मत कह देना फैकल्टी को जो पढ़ा रहा होगा कि हमें आइडिया नहीं दिया था. Right now I am just giving you the idea so how it is concerned and how it is related. कि अपन ऑक्सीजन डिसोसिएशन कर्व पढ़ेंगे तो उसमें किस तरह से अपन उसको कोरिलेट करेंगे कि कैसे ऑक्सीजन की बाइंडिंग होती है, पहले एक से होगी, फिर उसको प्रमोट करेगा, सेकंड से होगी, थर्ड से होगी, फोर्थ से होगी. So this type of interaction is known as heme interaction, which is beneficial for addition of oxygen to the hemoglobin. Now this hemoglobin has a dissociation curve. This dissociation depends on number of factors. The number of factors which determine the oxygen dissociation curve is, one of them is 2,3-diphosphoglycerate. What is this? This is a byproduct of metabolism of glucose which is present in the RBC. RBC does not have any nucleus, any organelles, but it does survive. So some metabolism has taken place or is taking place round the clock. Okay, so this is a byproduct of the metabolism taking place in the RBC, that is 2,3-diphosphoglycerate, and this increase in the concentration will result into the shift towards right, that is increase in the tremendous or at a fast speed of dissociation or liberating oxygen. Okay, just the opposite will take place. If there is decrease in the 2,3-diphosphoglycerate concentration, increase in the acidic concentration, when the person is exercising, what is getting? Excess of acid is getting formed. Okay, much of the energy is required by the muscles, much of the oxygen is required, so it will be towards the shift. Right will be towards the shift will be towards right. Okay, so these are the factors which is going to affect the oxygen dissociation curve towards right or towards left, depending on the requirement. Sit down. Okay, so this is one point which is related to this oxygen dissociation curve, or this is first example of derivative of hemoglobin, that is oxyhemoglobin. Oxygen loading takes place in the lungs, forms oxyhemoglobin, deoxyhemoglobin after oxygen diffuses into the tissue. Okay, this is concentration of diphosphoglycerate, deoxyhemoglobin. The lower one you can see DPG, it's written. Oxyhemoglobin, no as such DPG can be seen. There is difference in the structure of the hemoglobin molecule. Yeah, increase in the concentration of deoxy. Carboxyhemoglobin replaces oxygen and binds. So the affinity of carbon monoxide is 200 times more than the affinity for oxygen. Okay, in your AK Jain or in some other book, it might have been written 200 or 250 times. So please at least consult 2-3 books. आपको खरीदने के लिए नहीं कह रहे, but you just consult it. And every book is available in PDF form, so you need not to buy every book, but you just have to get it download. And I think you all are techno-savvy that much, so you can have the values and just correlate the values. Okay, so it has 200 times higher affinity than oxygen. This may be seen with heavy smokers. Carbaminohemoglobin bonds to carbon dioxide. Okay. Now see this carbon dioxide is getting bind with the NHCO2. Okay. Loading takes place in the tissue. Methemoglobin occurs when iron is oxidized to +3. Okay. If it has to carry oxygen, the iron has to be present in +2 state. In methemoglobin, what happens is it is present in +3 state. Okay, which does not allow the binding and dissociation and association of hemoglobin. In order for hemoglobin to carry oxygen, the iron must be present in +2 state. This occurs due to the deficiency of iron, methemoglobinemia. Okay. Glycated hemoglobin, सुना है word? नहीं सुना? प्री डायबिटिक पेशेंट्स जो होते हैं वो कराते रहते हैं, जिनके फैमिली हिस्ट्री होती है डायबिटीज की, ठीक है, वो जनरली ये कराते हैं ग्लाइसेटेड हीमोग्लोबिन, हीमोग्लोबिन A1C. Okay, in this chain the fragments of the chain binds with the glucose. They show the tendency of binding with this particular chain, binding of glucose. So they are done routinely for an investigation of prognosis or information regarding the diabetes. Okay, the most abundant form of glycated hemoglobin is HbA1c, which has a glucose residue attached to beta globin. Okay, this has a special property of binding glucose with the beta chain. Increased amount of glycated hemoglobins are found in patients with diabetes mellitus. Clear? This is an index to go for diabetes mellitus checker. These are the different types of hemoglobin. अभी तक अपन ने meth hemoglobin तक क्या पढ़ा था? Derivatives of hemoglobin. Now this is types of hemoglobin. So glycated hemoglobin क्या है? Type of hemoglobin. Okay. Adult hemoglobin, the major hemoglobin in humans, that is 2 alpha and 2 beta chains are present. Okay. A2, the first appears in 12 weeks after birth, a minor component of normal adult hemoglobin. 97% जो एडल्ट हीमोग्लोबिन होता है वो तो होता है A, और ट्रेसेस अमाउंट के अंदर होगा A2, less than 2 to 3% is A2. Majority of the adult hemoglobin comprises of alpha and beta chains. Fetal hemoglobin normally synthesized only during the fetal development. Now it has the strong affinity for oxygen because it has to take oxygen from the maternal circulation. Okay, so the nature is created in such a way that the fetal hemoglobin has a special affinity and it will have the capacity to take from the maternal circulation. But this fetal hemoglobin, as is present at the time of birth, but actually it decreases, and within 1 year the hemoglobin gets transferred into total adult hemoglobin type of. So fetal hemoglobin is normally not present in adult person. It has to be diminished by the age of 1 year. Okay. Now this is sickle cell anemia. Okay. इसके ऊपर MCQ आता है. Glutamic acid is replaced by valine at the end at the 6th position. Okay. What happens in the hypoxia condition? What is hypoxia? When there is decrease in the oxygen concentration at tissue level. So when there is hypoxia, what will happen? The sickle, the RBCs, okay, when there is problem in this valine and glutamic replacement has taken place, now what will happen? The RBCs, there will be formation of crystals. These small crystals, they are known as tectoid crystals, and when tectoid crystal formation takes place, it makes the RBC structure from biconcave to sickle shape. Okay, when it becomes sickle shape, it loses all the importance of the biconcave nature of RBC. So their lifespan decreases, the hemolysis nature increases. Clear? So what happens is basically there is change in the protein, plasma protein, amino acid, sorry, plasma protein not, amino acid sequence is changed. Then there is, in hypoxia, there is tectoid formation which results or changes the RBC shape into sickle shape, and this sickle shape loses all the merits or the positivity of the biconcave shape of RBC. So when it is coming through the circulation, it will lose its property and result into hemolysis. Okay. Sickle cell anemia is a genetic disorder that is characterized by the formation of hard, sticky, sickle-shaped red blood cells, in contrast to the biconcave shaped red blood cells found in normal individuals. Okay. This disease is caused by a mutation which results into the positioning of glutamic by valine. Okay. Now important is, जो सबसे ज़्यादा पूछा जाता है आपसे एग्जाम में, प्रैक्टिकल वाइवा में, वो पूछी जाती है इनकी रेंज, इसकी वैल्यूज, ठीक है, एंड व्हाट आर द कॉजेज फॉर देयर इज दिस चेंज इन द वैल्यू इन मेल, फीमेल और डिफरेंट स्टेजेस. Okay, adult male it is 14 to 18 gram per deciliter. If this is the range, then the average comes out to be 15.5 gram per deciliter. In case of adult females, it is 12 to 15.5 gram deciliter, which comes out to be 14. That is the average. Okay. At the time of birth, it is 23 gram per deciliter. So the RBC count is more at the time of birth. So we will correlate it later on. Okay. Just remember it. Clinically, 14.8 gram per deciliter is considered as 100% hemoglobin, means it is totally or 100% saturated with oxygen. मैंने बताया भी था कि आपकी ट्यूब के ऊपर भी मार्किंग्स है, एक जगह परसेंटेज स्केल है, एक जगह ग्राम परसेंटेज है. तो 760 मिलीमीटर मर्क्यूरी प्रेशर पे पार्शियल प्रेशर ऑफ ऑक्सीजन तो 100% हीमोग्लोबिन के साथ हीमोग्लोबिन को सैचुरेट कर. Okay, so clinically 14.8 gram per deciliter is considered as 100%. Okay, 1 gram hemoglobin when fully saturated combines with 1.34 ml of oxygen is an index of oxygen carrying capacity of blood. So normal is 21 ml% in males and 18.5 ml% in females, adult male and adult female. Now what happens to the hemoglobin when it is getting or when the life cycle of RBC gets completed? ठीक है, कितना लाइफ स्पैन होता है आरबीसी का? 120 days. गुड. फिर उसके बाद क्या होगा इसका? डिस्ट्रक्शन होगा. डिस्ट्रक्शन कहाँ पे होगा? स्प्लीन. बस स्प्लीन नहीं होगा, लिवर में होगा, रेटिकुलो एंडोथेलियल सिस्टम में होगा, सेल्स में होगा, ठीक है, डिफरेंट मैक्रोफेजेस में होगा. फिर क्या होगा? जैसे अपन ने स्टार्टिंग में पढ़ा था हीमोग्लोबिन को हीम और ग्लोबिन, वैसे ही इसका लाइफ स्पैन कंप्लीट होते ही क्या ब्रेक हो जाएगा? हीम और ग्लोबिन. व्हेन वी आर टॉकिंग ऑफ हीम, the iron will come out separately. What will remain is the tetrapyrrole or protoporphyrin ring. This protoporphyrin ring will open up, straighten up, will get converted into biliverdin, and this biliverdin when oxidized get converted into bilirubin, and this bilirubin will further circulation will go either to the GI system, resulting into the removal from the GI system, urobilinogen, stercobilinogen, heme or the iron content is again recycled, but some portion of iron is lost through the desquamation of the epithelial cells of GI lining, and some of the iron is lost in case of females through the menstrual jet. Okay, so partly the iron is getting lost in different forms, but rest majority of the iron is getting recycled. So that is the breakdown of hemoglobin. Okay, the globin portion is made up of amino acids basically. Okay, so these are again reutilized. That is a separate story, but broadly when we are talking, it is mainly concerned with the heme. And what happens to the heme? The iron gets recycled again, and what is remain is the protoporphyrin ring. Now this protoporphyrin rings open up, get straightened up, converted into biliverdin, and this biliverdin further gets converted into bilirubin, and that is further taken by the GI system and results into bilinogen, urobilinogen. In case of liver, it combines with the plasma albumin, forms albumin bilirubin. This is a complex, but still the clinician calls it free bilirubin. So it has a separate. Now this will enter into liver, and this will further take place. We'll talk about this in jaundice. That is a separate section. But right now, this is the thing which happens to the hemoglobin or destruction of hemoglobin, which is taking place after the RBC complete its life cycle of 120 days. Clear? हाँ या ना, पता तो होगा इतना खैर. Okay, they are engulfed by macrophages. Heme and globin are separated. Iron is removed from the heme and reused, stored as hemosiderin or ferritin. What was the difference between hemosiderin and ferritin? The iron content in hemosiderin is more. Hemosiderin has more concentration of iron. Transported in plasma by beta globulin as transferrin. ठीक है? Heme is degraded to a yellow pigment called bilirubin. Liver secretes bilirubin into the intestine as bile, urobilinogen or stercobilinogen. It is respectively removed. Globin is metabolized into amino acids, which are then released or reused via circulation. The tissue macrophage system or the reticuloendothelial system includes the phagocytic cells which are present in liver, spleen and lymph. Okay, hemoglobin broken plasma proteins and amino acids pool में चला गया. Bilirubin, iron, the bone marrow for formation of erythropoiesis, and bilirubin binds with the albumin. Plasma albumin forms unconjugated bilirubin, enters into the liver. This is the section which we'll see in jaundice or GI. Okay, this is the formation and destruction of hemoglobin. Okay.